Slide 1. This unit is one of a series of slide tapes in renal physiology prepared by the American Physiological Society. 2. The program begins with a review of prerequisite knowledge. Before you go any further, make sure you have completed the entry skills test provided in the study guide. 3. During the presentation, question slides will appear at intervals. Stop the tape to answer the question. Then advance to the next slide on which the answer will appear. Stop the tape again to check your answer. After you finish the unit, assess your understanding of the material by taking the post-test in the study guide. 4. This slide tape discusses the regulation of renal potassium excretion. Content expert, Dr. Fred Wright. 5. In this presentation, we will be discussing, first, distribution of potassium in the body. Second, factors influencing renal potassium excretion. Third, renal mechanisms contributing to potassium excretion. Fourth, nephron sites involved in potassium excretion. And fifth, regulatory elements. 6. First, we will deal with the distribution of potassium in the body. When you have completed this section, you should be able to 1. Describe the relation between potassium intake and excretion in the normal adult. 2. Specify approximate percentages found in urine, feces, and sweat. And 3. Calculate the approximate amount of potassium in the extracellular fluid compartment. 7. Potassium, symbolized by the letter K from its Latin name kalium, is a highly reactive metallic element. It is found in nature in salts or in ionized form and is an important constituent both of ourselves and of what we eat. 8. Normally, the dietary intake of potassium is equal to the rate of excretion. This balance between intake and excretion over time is necessary to maintain a nearly constant amount of potassium in body fluids. The average intake is between 50 and 150 milliequivalents per day, but may be lower or higher. Excretion is mainly through the kidney, about 80 to 90% or higher. About 10% is excreted in the feces, and less than 1% is lost in sweat. 9. Most of the potassium in the body is found in the intracellular fluid compartment. Only about 2% is found in the extracellular fluid compartment. 10. The normal concentration of potassium in plasma is between 3.5 and 5 milliequivalents per liter. Since the volume of the extracellular fluid compartment is about 1/3 of total body water, and total body water is about 60% of total body weight, the amount of potassium in the extracellular fluid compartment of a person weighing 70 kilograms can be calculated to be about 65 milliequivalents. Incidentally, this amount is about equal to the amount ingested and excreted each day. 11. Despite the relatively large amounts added to body fluids each day, and despite the very large amount of potassium in cells, regulatory mechanisms usually keep the concentration of potassium in plasma quite constant. 12. When plasma potassium does change, it usually changes in the same direction as cell potassium changes, and therefore reflects gains or losses in total body potassium. For example, with large losses of potassium in urine, as caused by diuretic drugs, in feces, as in diarrhea, or in sweat, as in heavy exercise, both intracellular and extracellular potassium decrease. 13. However, this is not always true, and a low plasma potassium might be present without potassium deficiency if potassium shifts from extracellular fluid into cells. This can occur when extracellular fluid becomes alkaline, as after bicarbonate ingestion, or in treatment of diabetic ketoacidosis, as after insulin administration. 14. The distribution of potassium, high concentration inside cells and low concentration in extracellular fluid, plays a critical role in determining the electrical behavior of excitable membranes. The resting voltage across muscle and nerve membranes is altered when the relation between intracellular and extracellular potassium is changed. 15. Rapid increases in extracellular potassium concentration may depolarize the membranes and cause increased excitability. Since membranes of heart muscle cells are among those with increased excitability, the consequences can be lethal. 16. Remember to stop the tape to answer questions and to check your answers. 17. 18. The second part of this discussion deals with factors influencing renal potassium excretion. You should be able to indicate in which direction two of three variables, potassium, sodium, or aldosterone, will change when one of these three variables has been increased or decreased. 19. Renal mechanisms operate to maintain potassium balance. The kidney is able to adapt to wide variations in potassium intake by varying potassium excretion as much as 10-fold from usual rates. 20. It is possible to separate factors that influence the rate of renal potassium excretion into at least 4 categories. First, total body potassium. Second, sodium reabsorption and excretion. Third, aldosterone. And fourth, acid-base balance. There are interrelations among these factors. The separation is made mainly for the purposes of clarity in discussion. The mechanisms of these alterations will be discussed later in the presentation. 21. The first factor influencing potassium excretion is total body potassium. For example, when potassium is added to the body by increased intake, the kidney increases its rate of potassium excretion so rapidly that very little increase in total body potassium occurs. 22. When potassium intake is stopped, renal potassium excretion decreases to very low rates. 23. When total body potassium is depleted due to continuing non-renal losses, as in feces or sweat, the kidney conserves potassium and helps to bring intake and total excretion into balance. 24. The second factor influencing renal potassium excretion is sodium reabsorption and excretion. For example, when sodium-containing solutions are infused intravenously, causing an increase in extracellular fluid volume, or when diuretic drugs are administered, causing a decrease in tubule salt reabsorption, both sodium and potassium excretion increase. 25. When sodium intake is stopped and sodium reabsorption is more complete, both sodium and potassium excretion decrease. 26. The third factor influencing renal potassium excretion is aldosterone. If the plasma concentration of the adrenal mineralocorticoid hormone, aldosterone, changes, renal potassium excretion changes. 27. The first way in which aldosterone levels can be increased is by the primary overfunction of the adrenal cortex, or by the administration of mineralocorticoid hormones. In situations where there is an initial increase in circulating aldosterone, potassium excretion is increased. At the same time, sodium excretion is decreased. 28. A second way in which aldosterone levels can be increased is by increased potassium intake. A high potassium intake may stimulate aldosterone secretion and lead to increased potassium excretion. Simultaneously, sodium excretion is decreased. 29. A third important stimulus to aldosterone production is a low sodium intake. Thus, a period of sodium deprivation may stimulate aldosterone production enough to enhance renal potassium excretion. The net effect of both sodium depletion and high circulating aldosterone on potassium excretion will be a balance of two oppositely directed influences, since high aldosterone increases potassium excretion and low sodium decreases potassium excretion. 30. When circulating levels of aldosterone are reduced, potassium excretion is reduced and sodium excretion is increased. 31. There are important interrelationships, therefore, among total body potassium, potassium intake and excretion, sodium intake and excretion, and aldosterone. As this slide indicates, the nature of these interrelationships is complicated and tends to be somewhat confusing. Let's review the factors one by one. 32. Increases in potassium intake are followed by increases in potassium excretion. Decreases in potassium intake are followed by decreases in potassium excretion. 33. In some situations where renal sodium reabsorption is decreased, sodium and potassium excretion increase in parallel. Also, it should be noted that acute administration of potassium salts results in an increase in both sodium and potassium excretion. 34. Increases in plasma aldosterone result in increased potassium excretion and decreased sodium excretion. In turn, increased plasma potassium and decreased plasma sodium are stimuli for aldosterone production. Decreases in aldosterone have the opposite effect. 35. Therefore, when sodium intake changes, conflicting factors influence renal potassium excretion. For example, when sodium intake is reduced, plasma levels of sodium are reduced. Subsequently, increased aldosterone enhances potassium excretion, whereas the decrease in sodium excretion produced directly by reduced sodium intake tends to diminish potassium excretion. 36. The final change in the rate of potassium excretion will be determined by the sequence in which the factors are changed and by the relative magnitudes of the changes. 37. In addition to the three factors already discussed, total body potassium, sodium reabsorption and excretion, and aldosterone, renal potassium excretion is influenced by a fourth factor, acid-base balance. Acute changes in acid-base balance leading to increased alkalinity of extracellular fluid result in increased rates of potassium excretion. For example, potassium excretion increases in severe vomiting or hyperventilation due to increases in alkalinity of the extracellular fluid. 38. Conversely, acute hypoventilation and its accompanying increased acidity of extracellular fluid results in a decreased rate of potassium excretion. 39. These changes in potassium excretion may be related to the shifts of potassium between extracellular and intracellular fluid that were discussed earlier. For example, extracellular alkalosis causes potassium to shift into cells and leads to increased potassium excretion. When changes in acid-base balance are prolonged, as in chronic renal or respiratory diseases, other factors appear to become important, and it is not really useful to attempt to predict the expected rate of potassium excretion. 40. 41. 42. 43. 44. In the third part of this discussion, we will consider renal mechanisms contributing to potassium excretion. Given the GFR, plasma potassium concentration, and the rate of reabsorption and excretion, you should be able to calculate 1. Rate of potassium filtration per day, 2. Milliequivalents per day of potassium reabsorbed, 3. Milliequivalents per day of potassium secreted. 45. The processes within the kidney leading to potassium excretion are filtration by the glomerulus and both reabsorption and secretion by the renal tubules. 46. Potassium ions in plasma are freely filtered. The amount of potassium filtered per day can be calculated from the glomerular filtration rate, about 150 liters per day, and the plasma potassium concentration, about 4.5 milliequivalents per liter, to be about 675 milliequivalents per day. 47. Potassium concentration in the urine may vary over a wide range. Since normal urine flow is about 1 liter per day and urinary potassium concentration is about 100 milliequivalents per liter, renal potassium excretion would be 100 milliequivalents per day. This is only an approximate value. Recall that it is within the range of average daily intake. 48. Potassium excretion can be expressed in several ways. One way is to calculate potassium excretion relative to potassium filtration. The ratio of these terms is the fractional excretion. Potassium excretion is normally only 10 to 20% of potassium filtration. A net quantity of potassium equal to more than 80% of the filtered amount is normally reabsorbed. 49. The fractional excretion can also be calculated in two other ways. 1. As the ratio of potassium clearance and inulin clearance, and 2. As the ratio of urinary and plasma potassium concentrations divided by the ratio of urinary and plasma inulin concentrations. 50. The difference between filtration and excretion is a minimum estimate of the amount of potassium reabsorbed. It is usually not a true estimate because, as we will see, secretion also contributes to excretion. The potassium that is excreted is not simply the amount of filtered potassium that escapes reabsorption. 51. For example, when kidney disease has reduced GFR to very low levels, it has been found that potassium can be excreted at a greater rate than it is filtered. 52. This observation from the diseased kidney indicates the existence of a process for transporting potassium from capillary blood into renal tubules, that is, a secretory process. The difference between potassium excretion and filtration minus reabsorption indicates a minimum value for potassium secretion. 53. 54. 55. Our fourth objective is to identify nephron sites involved in potassium excretion. Given a rate for fractional excretion of potassium, from a knowledge of micropuncture experiments, you should be able to specify the fraction of filtered potassium that remains at progressively more distal sites along the nephron. 56. Experiments performed to identify the sites where the reabsorptive and secretory processes are located have employed micropuncture techniques to collect samples of tubule fluid from proximal and distal segments of superficial nephrons. The amount of potassium remaining at a site along the nephron, the end of the proximal tubule, for example, can be expressed as a fraction of the filtered amount. This fraction is a dimensionless quantity. Measured values for potassium and inulin concentrations in tubule fluid are used instead of the concentrations in final urine. 57. Actual results of such measurements made in rats fed a low potassium diet and excreting about 0.05 or 5% of filtered potassium are shown. Of the total amount filtered and entering the proximal tubule, about 0.3 remains at the end of the proximal tubule. About 0.1 remains at the end of the loop of Henle and enters the distal tubule. About 0.07 remains at the end of the distal tubule, and 0.05 at the end of the collecting duct. These data show continuing net potassium reabsorption along each of the main nephron segments. 58. Results of similar measurements made in rats fed a high potassium diet and excreting more than the filtered amount of potassium are shown. Here again, the amount remaining at the end of the proximal tubule is about 0.3 of the amount filtered, and the amount entering the distal tubule is again about 0.1 of the amount filtered. In this situation, however, continued reabsorption is not seen along the distal nephron. The amount of potassium in samples taken from the end of the distal tubule, 1.2, was greater than the amount entering the distal tubule, and was even greater than the amount filtered into the nephron at the glomerulus. 59. These and other data show that potassium excretion can be attributed largely to potassium secretion. The data also show that reabsorption occurs mainly in the proximal tubule. The distal tubule and collecting duct are the sites of potassium secretion and are responsible for varying the rate of potassium excretion in response to changes in potassium intake. 60. Neither the rate of glomerular filtration nor the reabsorption by the proximal tubule and the loop of Henle have much effect on the rate of potassium excretion. Some additional reabsorption may normally occur as the tubule fluid passes through the terminal portion of the collecting duct. 61. It has also been observed that the distal nephron's variable capacity for potassium secretion is responsible for the increased rates of potassium excretion that occur, 1. When total body potassium is increased, 2. When sodium excretion is increased, 3. When aldosterone is increased, or 4. During the early stages of metabolic or respiratory alkalosis. 62. 63. 64. In the last portion of this discussion, we will consider regulatory elements. You should be able to, 1. Name the 2 steps involved in the transport of potassium from peritubular blood to the lumen of the distal nephron and specify the driving forces for these transport processes. 2. Specify the 2 main determinants of the rate of potassium secretion by cells of the distal nephron. 65. There are 2 initiating factors that set the rate of potassium secretion by cells lining the wall of the distal nephron. 1. Factors driving potassium into cells from the peritubular capillary blood, and 2. Factors pulling potassium into the tubule from the cells. 66. Potassium is transported from blood to tubule through cells of the distal nephron. The potassium concentration in these cells, as in other cells, is 10 to 20 times the concentration in plasma and interstitial fluid. For the first step in secretion, the entry into cells, potassium must move uphill against an electrochemical gradient. It depends, therefore, on active transport, or pumping. This active accumulation of potassium maintains cell potassium at a high concentration. 67. The force driving potassium out of cells is determined by the intracellular concentration and by the voltages across the cell membranes. Since the voltage across the luminal membrane is less than the voltage across the peritubular membrane, the luminal barrier offers less opposition to potassium flow out of the cell. 68. Net secretion of potassium may thus be explained as involving 2 transport steps: an active uptake from blood to cell and a passive leak from cell to lumen. A high rate of active uptake would tend to increase cell potassium concentration and thus drive potassium across the luminal membrane at a faster rate. 69. Fluid entering the distal tubule from the loop of Henle has a low potassium concentration. This fluid carries away potassium that has been transported into the lumen. Usually, the concentration of potassium increases in the tubule fluid along the length of the distal tubule. 70. The rate at which potassium concentration increases with length is affected by the relation between potassium transport rate and volume flow rate. Of a variety of theoretically possible relationships between the rates of ion transport and the rate of volume flow, 3 cases of particular interest will be discussed. 71. Case 1. If the potassium transport rate is constant, increases in volume flow would diminish the rate of increase of potassium concentration with length. 72. Case 2. If volume flow is constant, increases in potassium transport would increase the rate at which potassium concentration rises along the tubule. 73. Case 3. If the potassium transport rate increases in proportion to increases in volume flow, there would be no change in the rate at which potassium concentration rises along the tubule. 74. Data from micropuncture experiments show that the behavior of the distal tubule usually conforms to either the second or the third case. In some situations when potassium excretion is increased, distal potassium concentration is increased. Examples of case 2 are ingestion or infusion of potassium salts, administration of mineralocorticoid hormones, and acute alkalosis. It appears that the main determinant of the increased rate of secretion is initiated by an increase in peritubular uptake of potassium and therefore an increased flow of potassium from cell to lumen. 75. In other situations, when potassium excretion is increased, distal potassium concentration does not change very much. Examples in case 3 are acute infusion of sodium salts and administration of diuretic drugs that inhibit salt and fluid reabsorption in the proximal tubule or the loop of Henle. As a result of decreased salt and water reabsorption in the more proximal parts of the nephron, a primary increase in volume flow into the distal tubule initiates first a proportional increase in the potassium transport rate, and second, increased uptake of potassium from blood. 76. We have discussed 2 processes, potassium secretion and potassium excretion. Potassium secretion takes place in the distal nephron. It is initiated by factors driving potassium into cells or by factors pulling potassium into the renal tubule. The rate of potassium excretion is determined by the relationship between the rate of secretion and the rate of volume flow. 77. Finally, in situations where potassium excretion is decreased, such as when potassium intake or sodium intake is reduced, potassium secretion by the distal nephron is turned off, and both sodium and potassium are reabsorbed. In the case of sodium depletion, potassium reabsorption occurs even though circulating aldosterone is increased, and indicates that additional factors not yet well understood can overcome the effect of aldosterone and promote potassium reabsorption in the distal nephron. 78. 79. 80. This concludes the discussion of regulation of renal potassium excretion. Refer to your study guide for the post-test and references for additional reading.